You have the floor. Thank you very much. Good afternoon. Oh, what he didn't tell you is that my background, I started working with poultry production. And now I'm going to talk about this topic applied to ruminants. So I'll do my best to discuss this topic with you. How can we have this link between intestinal health, immune response, and production? When we talk about intestinal health, when we talk about ruminants, we have to start thinking about the four initial compartments of the GI tract. Nutrition, all the factors are being analyzed. And the GI tract, the lower part where we have the intestines. So now let's have a magnifying glass on this part here of the intestine. This tube, if we have a magnifying glass on it, this is what we are going to see, the different tissues, the villus, and then I will be able to see the villi, and I'm able to see cells and all the structures. And all of this that we invest in production is to get to the end over here in terms of protein and nutrients that have to be absorbed. So maintaining the integrity of the GI tract is essential so that the investment that we are making will be used by the animal. We have some microscopic pictures. They are all villi. We can see some bacteria that have been adhered, beneficial bacteria that are adhered to the epithelium. Okay, so this is what we are talking about. And what is the relationship between intestinal health and performance? That is a very close relationship between the brain and the intestine. This is known now as the Microbiota-gut-brain axis, and where we have an additional factor because we understand the importance of the microbiota and how it's related to intestinal health and even metabolic response. Very briefly, we have an example here, a very practical example. When the animal is in an environment of heat stress, nothing that is affecting the gut, no, it's an environmental factor. It starts to trigger responses from the brain. Some hormones are produced like cortisol. And the intestine is one of the organs affected by cortisol. And on the long term, this can affect permeability of the velocities and also affecting the intestinal microbiota. The opposite can also happen. And this applies to us as well, not only animals. If there is some anti-nutritional factor, a contamination by a pathogen, this will alter the microbiota, the intestinal permeability, and there is also the production and release of certain components that will communicate with the brain. This relationship can be measured in several ways, but that is direct and is both ways. It can be started by either one, either the brain or the intestine. When we are talking about this, there is a direct interaction between brain and the intestine and microbiota and vice versa. Here I have an example of how we talked about this relationship. So when there is an infection, wherever it occurs, we know that there is a mobilization of nutrients for this response. And I brought you this example here, an example to have a better response. So we have an infection process here. If there is a weight loss due to this infection, we are able to have a partitioning of this metabolic availability. 68% is a decrease in intake, feed intake, and the rest, the remaining 5%, comes from the activation of the pro-inflammatory response and the activation of the immune system cells. 3.5% is involved in the increased metabolic rate. And on the other hand, we have redistribution of nutrients. Growth is no longer a priority. The inflammatory process is the priority. And 9% is related to the intestinal epithelium, damages that can occur, or they can be a consequence of the metabolic process, and losses. When we have damages to the intestinal epithelium, we don't have the nutrients absorption. We also have a metabolic expenditure to cover the damages, produce new cells, have a cellular turnover, etc. So this is the way these expenses, this occurs, whatever needs to be repaired. Another example, we are talking about the energetic cost. This happens also in humans. This is a human study. In an inflammation, the additional energy cost can be increased by 25 up to 60%. Low-grade chronic infection, 10%, acute pain, up to 60% or more. So for us to understand that this is going to happen when we have an inflammation, an infection. And what about the amino acid requires? This is monogastric in swine, and here we have the demand of amino acids for protein accretion in the body. So we will always formulate the diet based on all the nutritional requirements. And these requirements were established, but didn't take into consideration the stress factors. It's just for accretion, protein accretion and growth. What happens with some of the specific amino acids when we have inflammatory response and infections? The C-reactive protein works against the pathogens. EGA is an immune globulin, and mucin is the mucus that protect the mucosas. They are all protein-based. Some of them, like threonine and valine, they have their requirements very often almost 8 times higher. So besides the metabolic priority that the immune system has in a pro-inflammatory process, the nutritional demands are totally different on the case. So we need to have this in mind. And now to close this topic, the energy demand for some tissues, some organs, are much higher than for protein accretion. And the priorities, they will always be met before the other requirements. So the second message that I would leave, the immune system is the priority in nutrition partitioning and demands a high metabolic cost, both in energy and amino acids. With these two concepts in mind, let's talk now about immune nutrition. It was started with human nutrition, with human medicine in the 50s, when populations were started to be studied and understand how the, the food consumption in one population would affect public health and diseases. So up to the 70s, this concept of human immunonutrition was started. And in the last 20 years, this started very slowly in animal nutrition, but it is fully applicable. I have here a definition of a publication in human studies. Immune nutrition is the potential of modulating the immune system activity by intervention with specific nutrients, certain components. It was started with nutrients that are going to be absorbed, but now we know that the microbiota is another factor. So everything that can modulate the microbiota and can interact with the epithelium and the membrane in the intestine will be included here. And this is what I bring you now. With immune nutrition concept, we have immune nutrients. And this nutrient or this component needs to have an effect on the integrity of the intestinal barrier, on the cell defense function, and also an effect on the systemic inflammatory response. In all of them, in one or more of them, this is considered to be immune nutrients. So we have the classical ones that I mentioned, threonine, that has a high demand for mucus production, IgA. But we have others like glutamine, that is very well known as a source of energy for the cells that are undergoing multiplication in the GI tract. We also have some vitamins. Sometimes we know this better in human nutrition than in animal nutrition. Some minerals, selenium, iron, zinc, lipids, the nucleotides, when they are free in the diet, they can also be absorbed in the intestinal. When we talk about an adult ruminant, it has to be protected to reach the intestine. In a young animal, it can be absorbed in the intestine, and it will facilitate the absorption. And we have additives, those that are not nutrients by themselves, but have a potential to modulate the microbiota, to improve the immune response, the barrier of the mucosa. So this is the link that we establish, and I'm going to bring to our discussion at ICC the yeasts. Yeasts cell wall are very high in some carbohydrates, functional carbohydrates, beta-glucans. And I bring to you just an image. I don't know if during the pandemic, there are some fridges and they have supplements. And there were many of these supplements became available. The same concept that we talked, some vitamins, some amino acids, and beta-glucan from yeasts. And they are here in the composition of this supplement based on whey. Why is it that the beta-glucans are considered to be an immune nutrient? When we talk about beta-glucans and they reach the gut, here we have an intestinal epithelium. Below it, we have a tissue that is called lamina propria. where we have a large number of immune system cells, namely the innate immune system. They stay here and they stretch an arm. They are not loose in the intestinal epithelium, but they are stretching their arms to have contact in what is close to the epithelium. Their function is to know if there is some threat, capture, phagocyte it, and present it to the immune system, saying, hey, we have to promote an immune response. So these cells, macrophages, dendritic cells, lymphocytes, they are known as antigen-presenting cells. They are the first line of defense, identify what it is, and we are going to start the response. What happens with the cell wall of yeasts? They will go very close here. They are recognized by the cells. Here we have a better drawing. They will be recognized as these cells have receptors on their membrane recognizing pathogens, antigens, etc. The next step is to have phagocytosis. This is the way to inactivate the pathogen, as it is the first defense line. Break it down in smaller pieces to recognize them and release this stimuli to the immune system, saying there's something here that we have to pay attention to. Pro-inflammatory cytokines are released and some other stimuli to produce a specific immune response like immunoglobulins, antibodies, and beta-glucan is very well known. There are many studies showing their potential of being an immune modulator. They are recognized by these cells. They have this initial process, recognition and recruitment to increase the cells of the defense line and not have a high response as if it were a pathogen. Beta-glucans are not multiplying, they are not causing any damage to the epithelium, it's not invading the epithelium. So with this response, it is already enough to control them. And when we have this increase of the response, when we have a true pathogen, a challenge, the immune response is already one step ahead and is able to respond. So this is the immune modulator effect of beta-glucans. To show this, I have here some results of studies in calves and in beef cattle, in cows, and I try to focus in heifers and in beef cattle. This was a study that was carried out in the state of Paraná, to Professor Heloísa, Professor Neumann. A part has already been published. What was done in this study? 36 Angus animals that were placed in the feedlot and went through a 105 days period in the feedlot. We divided them into 3 groups. One is the control group, and the other is 2 levels of rumen yeast that William is going to talk about, but it has mannano, oligosaccharides, and beta-glucans. We had the supplementation and blood samples were collected on days 0, 14, 42, 70, and 108. What was measured? We have several immune responses that were measured in this study. But here we can see the oxidative neutrophil metabolism. Neutrophils are cells of the innate immune system. They are also antigen-presenting cells, so they are able to respond. What did we see? That around days 42 and 70, there was a climate change. We were not able exactly what was the day, but all the responses show that there was a climate challenge. October, November, December, very hot during the day. It rains at the end of the day, so then we have thermal inversion. Animals had nasal secretion, etc. What did we see here? This response, when we measure the metabolism of these cells, it shows that they are more active. And this is a way to identify the level of response of the innate immune system. On the day 70, when we had this challenge, the 2 doses of rumen yeast that were tested were significantly superior in relation to the innate immune response. But then you can ask me, okay, but there is a higher energy and metabolic expenses. Yes, but what effectively happened at the end? There was a lower incidence of nasal secretion and lower alteration in the body temperature of these animals. Professor Eloisa is going to bring to you more details about these studies and talk a little more about the immune response. But what we are looking for is to give the animal a tool to be prepared to challenges. It's not going to solve it, but it's going to help the animal to pass by this challenge and to have more nutrients to overcome the challenges. Another study here with heifers, they started to be supplemented with rumen yeast. We had a control group and a supplemented group. They started to be supplemented on day 15, if I'm not mistaken, and on day 30, they received a vaccine against the bovine respiratory complex. It causes a reaction. Animals have nasal secretion, signs of pneumonia, etc. But we do this to protect the animals, right? So we collected blood samples after days 33, 36, and 51. And in IgA was then quantified as we expect the animals to produce them and as it will act on the respiratory mucosa. And there was a statistical difference in IgA production circulating in the blood of animals that were supplemented with rumen yeast. This is an immunomodulator effect that will also be reflected in the humoral response faster and more consistent after challenge. What resulted? A lower pneumonia score. There were other items as weight gain, but this was the main objective of the study. Another component that we have, another functional carbohydrate, this is MOS, mannano oligosaccharide. An example of how a pathogen or a bacteria, a E. coli or salmonella, adheres, attaches to the intestinal surface. This bacteria has fimbriae that will adhere to the glycocalyx surface, and then it is able to multiply, produce toxins that will rupture the cell membrane and the tight junctions. When we have MOS, the same bacteria with fimbriae, they will attach to the carbohydrate instead of adhering to the surface of the intestine. Doesn't happen 100% of the cases, but this is an important point as we have carbohydrates that are not digestible and they will bring this bacteria with them. This is the mode of action of MOS. When we talk about MOS, we say that it's characterized as a prebiotic, but the classical classification is a fiber that is going to be fermented by the beneficial bacteria. But as they are with the beta-glucans, the wall becomes very indigestible. So it is present on the surface and they will be carried out of the body with the feces. So talking about this, I have here two results of the same study that I mentioned about the test by Professor Eloisa. At 105 days, there was a reduction of E. coli burden, statistical, in the feces of these animals that were supplemented with rumen yeast. And this also had an effect on time when you compare days 28 and 105. This other study that was made with lambs, we had, I'm not going to talk about the details, we had this E. coli strain was statistically reduced when rumen yeast was supplemented or when it was added to live yeasts. It's very different. Its purpose, its functionality is very different. So reduction of the contamination to show that there is less damages to the epithelium and less inflammatory processes. So it has an impact on food safety. And translating this for intestinal health and performance with the heifers, the response in EGA, these heifers had a myria contamination, so we already expected these animals to have diarrhea. And we saw that there was a decrease in the number of diarrhea episodes comparing the control and the supplemented groups. The animals that were supplemented didn't have 3 episodes, and the duration of the diarrhea was also decreased. And what happened in terms of weight gain? As of the 3rd week, we already started to see weight gain that was statistically different between the groups. So here we are talking about health, intestinal health, but this is also important in relation to the immune response to the intestinal health. So when we talk about MOS, beta-glucans, this is very important and will be reflected in the performance. This is a study from João at the UNESP. These animals, during the grow-out period, they were on pasture. They were not supplemented with rumen yeast, but by immuno-wall, only beta-glucans and MOS. This supplementation took place during 208 days, animals on pasture. Samples of the jejunum and ileum were collected, and there was an increase in the perimeter and in the area of the crypts and the villi. When we talk about the villi, when I have the depth is when I'm talking about the crypt. This is where the cells multiply, where the cell multiplication starts to repair damage of the epithelium, for example. This crypt can be increased by the challenge. On the other hand, the crypt, if the crypt is larger, it can be related to a height of the villi and also the area of the intestine that is healthier. This was also reflected in a higher daily weight gain, 29 grams per day of these animals. And I would like to bring the final messages. I'm not talking about cost. There are many other factors to be understood. But what is really important about this topic is that there is a direct interaction between brain and intestine plus microbiota and vice versa. The immune system has is a priority in the partitioning of nutrients and demands a high metabolic cost. Immune nutrition is focused on strengthening the immune response when the animal is under challenge. But this world without challenges does not exist. The immune modulator effect that we saw with beta-glucans, we have to train the innate immune system, and this is reflected in humoral response that is faster and more consistent. Maintenance of the health and integrity of the intestine with less metabolic costs and positive impacts in performance. Sometimes it's difficult for us to see this. We start thinking about calves, where it's more clear than in adults, but an adult animal in feedlot or on pasture, we have alterations that can be remediated with this concept. This is what I had to bring to you. I thank you very much, and we can talk later if you have any doubts.